Myself Professor Shamsul Hayat from Aligarh Muslim Liga. Today we will discuss Phytochrome, which is one of the lectures of course Plant Physiology and Metabolism. Before starting it, firstly we will shed some light on photomorphogenesis. While we often associate light with photosynthesis, it also plays a pivotal role in the overall development of plants. An interesting phenomena occurs when seedlings are grown in the absence of light. They exhibit what we call etiolated growth. This distinctive appearance features pale, tall and spindly seedlings. Beyond photosynthesis, light induces change in the plant growth and development, and we refer to this as photomorphogenesis. The term photomorphogenesis is derived from Latin words meaning light and formation. Light serves as signal that triggers and regulates these developmental changes. Among the key photoreceptors involved in these responses are phytochrome, cryptochrome and phototropin. In this lecture, our focus will be on phytochrome. Phytochrome, which is a blue protein pigment with a molecular mass around 125 kilodalton, serve as a light detector in plants as well as in certain bacteria and fungi. The discovery of phytochrome is closely linked to research on flowering. The term phytochrome, meaning plant color, was initially coined to refer to the protein pigment governing photoperiod detection and floral induction in specific short-day plants like cucumber and soybean. In 1932, the Bestville research group of the USDA, led by Borthwick and Hendricks, demonstrated that red light, that is 630 to 680 nanometer, prompts lettuce seed germination, while far red light, that is 710 to 740 nanometer, hinders the process. Seeds exposed to red light at the final treatment exhibited nearly 100% germination, whereas those exposed to far red light at the end experience a significant inhibition of germination. Phytochrome can interconvert between PR and PFR forms because phytochrome exhibits a unique characteristics. It has two distinct chemical structures that can transform into each other. These forms names are based on the color of light. They most effectively absorb PR, a blue form, absorb red light having wavelength 660 nanometer, while PFR, a blue-green form, absorbs far red light of wavelength 730 nanometer. Moreover, these pigments are set apart as they absorb photons and undergo a chemical conversion into the opposite form. The absorption spectra of the two phytochrome forms differ. PR strongly absorbs red light, whereas PFR strongly absorbs far red light, that is 730 nanometer. Both forms exhibit some absorption in the blue part of the spectrum. These differences in absorption are rooted in the distinct chemical structures of PR and PFR. Photoreversibility, which is photoconversion, also referred to as photochromism, is a defining feature of phytochrome. As you look, phytochrome are synthesized as an inactive red light absorbing isomer called PR. Red light absorption by PR form converts it into an active far red absorbing isomer called PFR. Absorption of far red light by PFR form converts it back to PR form. Thus, red light promotes phytochrome responses by converting inactive PR into active PFR form. Far red light inhibits the red light induced response by converting PFR back to PR spontaneously as well. Such a spontaneous reversion of PFR to PR occurs both in light and dark. Since it is measured by a decrease in PFR concentration, it is referred as dark reversion. Phytochrome pool is never fully converted to PFR or PR form because the absorption spectra of PFR and PR forms overlap. When PR molecules are exposed to red light, most of it is converted to PFR, but some of PFR is spontaneously converted back to PR. The proportion of phytochrome in PFR form after providing saturating red light condition is about 88%. Likewise, a very small amount of far red light absorbed by PFR form leads to incomplete conversion of PFR to PR. Thus, an equilibrium of 98% PR and 2% PFR is established. This state of equilibrium is termed as photostationary state. Photoconversion of phytochrome provides assessment of the available red to far red light ratio in the plant's environment. Low RFR ratio has a variety of development effects on various species. It inhibits seed germination in Arabidopsis and induces shade escape responses during plant growth. Plants growing under a canopy use phytochrome to sense RFR ratio so as to regulate shade avoidance, seed germination, etc. In natural conditions, plants are exposed to a broad spectrum of light. RFR ratio is strongly affected by plant canopy because chlorophyll absorbs red light but not far red light, thereby affecting RFR ratio for plants growing below the canopy. Now let's discuss about the structure of the phytochrome. Phytochrome is a soluble protein with a molecular mass of about 250 kilodalton. It occurs as a dimer made of two equivalent subunits. Each subunit consists of two components: a light absorbing pigment molecule called the chromophore and a polypeptide chain called the apoprotein. The apoprotein monomer has a molecular mass of about 125 kilodalton. Together, the apoprotein and its chromophore make up the holoprotein. Moreover, in higher plants, the chromophore of phytochrome is a linear tetrapyrrole termed phytochromobilin. There is only one chromophore per monomer of apoprotein, and it is attached to the protein through a thioether linkage to a cysteine residue. As you can see in the figure, on absorption of light, the PR chromophore undergoes a cis-trans isomerization of the double bond between carbon 15 and 16 and the rotation of the C14 and C15 single bond. Assembly of the phytochrome apoprotein with its chromophore is autocatalytic, that is, it occurs spontaneously. Just have a look at the figure. Light absorption by phytochromobilin leads to its isomerization, which results in changes in its absorption spectrum and conformation of the phytochrome protein. The phytochrome protein consists of an N-terminal photosensory region and a C-terminal regulatory domain. The two are attached with the hinge between them as illustrated in the figure. The photosensory part has four domains, that is P1, P2, P3 and P4. P1 and P4 function in the inhibition of dark reversion of PFR to PR. P2 acts as a PS type of domain which is involved in sensing of signals, and P3 is the binding site for the phytochromobilin molecule. The regulatory region of phytochrome protein consists of two PAS type domain. Moreover, PAS, that is per AR sim, refers to some alpha beta conserved structural domain in a wide range of prokaryotic and eukaryotic proteins involved in sensing of signal. C-terminal domain of the regulatory region exhibits kinase activity. Phytochrome responses are distinguished by the amount of light required to induce them. The amount of light is referred to as the fluence, the number of photons impinging on a unit surface area. The standard unit for fluence are micromoles of quanta per square meter. Some phytochrome responses are sensitive not only to the fluence but also to the irradiance or fluence rate of light. The units of irradiance are micromoles of quanta per square meter per second. As you look in the figure, there are three types of phytochrome responses based on their sensitivities to fluence. They are very low fluence or VLFRs, low fluence or LFRs, and high irradiance response, that is HIRs. Now we will discuss each of them. Coming to the VLFRs, they are non-photoreversible and can be initiated by fluencers as low as 0.0001 micromole per meter square, and they saturate at about 0.05 micromole per meter square. The minute amount of light needed to induce VLFRs convert less than 0.02% of the total phytochrome to PFR, because the far red light that would normally reverse a red light effect converts 98% of the PFR to PR as discussed earlier. About 2% of the phytochrome remains as PFR, significantly more than is needed to induce VLFRs. Thus, far red light cannot reverse VLFRs. Coming to another phytochrome response, that is LFRs. Unlike VLFRs, LFRs are photoreversible and initiated at the fluence rate 1 micromole per meter square and are saturated at about 1000 micromole per meter square. These include most of the far red photoreversible responses, such as the promotion of lettuce and leaf movement. LFR spectra include a main peak for stimulation in the red region, that is 660 nanometer, and a major peak for inhibition in the far red region, that is 720 nanometer. Both VLFRs and LFRs can be induced by brief pulses of light, provided that the total amount of light energy adds up to the required fluence. Third response, that is HIRs. They require prolonged or continuous exposure. To light of relatively high irradiance. The response is proportional to the irradiance, loosely speaking the brightness of the light rather than to fluence. HRs saturate a much higher fluences than LFR, at least 100 times higher. The examples can be provided for the very low fluence rate, such as in the case of Arabidopsis seeds, which can be germinate with very low red light. The reciprocal relationship between fluence and time, known as the Bunsen-Roscoe law of reciprocity, is valid in case of VLFRs, which however fail to show reversal control by light. This means that a response can be induced either by brief pulse of red light that is quite bright, or by a very dim light for a longer duration. Other point of interest is that far red cannot reverse VLFRs. The reason is that about 3% of the total phytochrome remaining after far red exposure is sufficient to induce VLFRs. As you look, low fluence responses are the most studied changes which regulates plant growth and developmental processes like deethylation and seed germination. In deethylation, green expanded cotyledons with no apical hook and a short hypocotyl emerge from the soil and begin to become autotrophic. While in seed germination, it can be initiated or inhibited depending upon the species. Breakage of seed coat and activation of metabolism in plants are the few response of LFRs. This slide discusses high irradiation response. Some photomorphogenetic responses requires prolonged or continuous exposure to light of high irradiance or proportional to irradiance, but the reciprocity law is not followed here. Examples of HIRs are anthocyanin synthesis in dicot seedlings and apple skin, inhibition of seedling elongation, that is hypocotyl, flower induction, plumular hook opening, cotyledon expansion in mustard, ethylene production to sorghum, deethylation of seedlings. However, the effect is not photoreversible. The reason that these responses are called high irradiance response rather than high fluence response is that they are proportional to irradiance, that is brightness of source, rather than to fluence. This slide shows the types of phytochrome. Phytochrome is encoded by a family of divergent genes that give rise to two types of protein, that is type 1 and type 2. Type 1, which is encoded by the PHYA gene, is abundant in etiolated tissue. However, type 1 phytochrome is present at low levels in light grown plant because of its instability in the PFR form. The Phy-A mediated suppression of transcription of its own gene and the instability of its mRNA. Type 2 phytochrome, which is encoded by the PHYB, PHYC, PHYD and PHYE genes. It is present at low levels in both light grown and dark grown plants because its genes are constitutively expressed at low levels and the protein is stable in the PFR form. This slide basically discusses the activation of gene expression. Phytochrome regulates transcriptions of numerous genes and the stimulation and repression of transcription by light can be very rapid with lag times as short of 5 minutes. Many of the genes involved in greening, such as the nuclear encoded genes for the small subunits of rubisco and the chlorophyll AB binding protein of the light harvesting complex, are transcriptionally regulated by phytochrome, that is both phytochrome A and phytochrome B. As you can look in figure, PFR moves to the nucleus after absorption of red light. Two nuclear proteins that interact with phytochrome have been identified to date, although there are probably additional targets. The first phytochrome interacting factor 3, that is PIF3, reacts with the C-terminal end of phytochrome A or phytochrome B. However, it reacts preferentially with the full-length phytochrome B protein in a light-dependent manner, and it is thought to be a functional primary reaction partner for this phytochrome, although its precise function is not yet known. PIF3 resembles transcription factors that bind to a particular element in plant promoters, that is G-box motif, that confers light regulation to genes. It is also known that phytochrome B in the PFR form can form a complex with PIF3 bound to its target DNA. A picture is therefore emerging in which some phytochrome regulated genes are activated directly by movement of phytochrome B to the nucleus in the PFR form. Once in the nucleus, phytochrome B interacts with transcription factors such as PIF3. A model for the direction activation of gene expression by phytochrome B in the nucleus is shown in the figure. Now we discuss about the mode of action of phytochrome, which involves a balance between negative regulators such as phytochrome interacting factors or PIFs, constitutive photomorphogenic 1 or COP1, and suppressor of phytochrome A105, also called ESPA1, which can inhibit phytochrome function, and positive regulator HY5, which enhances and facilitates phytochrome mediated processes in plants. However, elongate hypocortical 5 or HY5 operates as a positive regulator, enhancing the effects of phytochrome and promoting its mediated responses, contributing to favorable outcomes in plant development. This balance between negative and positive regulators governs the complex functioning of phytochrome in various plant responses to light stimuli. Let us discuss the sequential mode of action in the development of photomorphogenesis unfolds through a series of steps primarily governed by COP1. In the dark, COP1 is predominantly located in the nucleus. However, upon exposure to light, COP1 undergoes exportation to the cytoplasm. Within the nucleus, COP1 directly interacts with ESPA1, leading to the promotion of phytochrome A or phytochrome A destruction. The COP1 ESPA1 E3 ligase complex also plays a crucial role by ubiquitinating and facilitating the proteasomal degradation of proteins that promotes photomorphogenesis, including the BZIP transcription factor HY5, LAF1 and HFR1 as displayed in the figure. As light becomes available, COP1 gradually moves out of the nucleus to the cytosol, reducing its interaction with many of the transcription factors localized in the nucleus. This allows these transcription factor involved in the mediation of photomorphogenic development to bind to promoter elements within genes. This binding initiates a cascade of events contributing to the regulation and progression of photomorphogenesis in response to light stimuli. There are several physiological responses regulated by phytochrome. They are deethylation, shade avoidance, circadian rhythm, seed germination, and stem elongation. This slide discusses about the deethylation that is regulated by the phytochrome. Ethylated, that is typical seedling response when dark grown. Long hypocotyl, that is stem below the cotyledons. Not green, that is no chlorophyll, no chloroplast. Little leaf development or unfolding. Exposed to light, that is deethylated. Hypocotyl stops growing, chlorophyll and chloroplast develop, leaves unfold. Therefore, plants need light to developmentally progress from heterotrophic to autotrophic organism. Seeds can be of the two types, that is photoblastic positive, that is germination in light need high PFR-PR ratio, and negatively photoblastic, that is germination in light need low PFR and PR ratio. Now, we will discuss in detail about shade avoidance response. It is an important function of phytochrome which enables plants to sense shading by other plants. Plants that increase stem extension in response to shading are said to exhibit a shade avoidance response. As shading increases, the RFR ratio decreases. The greater proportion of far red light converts more PFR to PR, and the ratio of PFR to total phytochrome decreases. When stimulated, natural radiation used to vary the far red content. It was found that for so-called sun plants or plants that normally grow in an open field habitat, the higher the far red content, that is lower the PR total ratio, the higher the rate of stem extension, as you can see in the figure. However, in other words, stimulated canopy shading, that is high levels of far red light, induce these plants to allocate more of their resources to growing taller. This correlation did not hold for shade plants which normally grow in a shaded environment. Shade plants showed little or no reduction in their stem extension rate as they were exposed to higher red far red values as shown in the figure. Thus, there appears to be a systematics. This slide shows the light quality that has a direct effect on plant growth via phytochrome. Consider bean plants given an end of the day red or far red light treatment. Plant growth is inhibited by red light, then higher PFR. FR light has no effect in etiolated seedling, that is high PR. The effect of phytochrome is thus developmentally dependent. FR actually stimulates stem growth at high fluence rate in green plants or inhibit it. Contrast this observation with the germination of lettuce seedlings. All of the previous studies were performed in the lab. What happens in the real world? Under a forest canopy, there is a little available red or blue light which are absorbed. Lots of FR, that is chlorophyll transparent, low RFR ratio. So, this converts PFR into PR. This suppresses lengthening of internodes. Thus, the relative amount of R and FR modulates the production of the active form of phytochrome and thus the metabolic activity in the plant. Transcription of genes needed for growth and enzyme activity. So, we conclude here as phytochrome plays a vital role several developmental processes in response to light stimuli. Its unique ability to exist in two interconvertible forms, that is PR and PFR, allows it to perceive a specific wavelength of light, facilitating intricate signaling pathways. The balance between positive regulators like HY5 and negative regulators such as PIFS, COP1 and SPA1 intricately modulates phytochrome's action, influencing plant growth, flowering and other essential responses. The dynamic interplay of phytochrome with downstream effectors and regulators unveils a captivating mechanism that adapts plant development to changing light conditions. Understanding the phytochrome functions opens new opportunities for manipulating plant growth and responses advancement in agriculture and horticulture. So, this is all about the phytochrome. Thank you so much.